Okay, so I'm Taina Pennanen, and maybe now it's fair that because Krista was talking about agricultural soils and Jenny about peatlands, and now I'm talking about forest soils and microbes, because the 70% of the land of Finland is covered by forests, we do have even though we do have altogether like 33 species, but 90–70% of the area is covered by 3 species: Scots pine, Norway spruce, and birch. Because of the ice age, proximity of ice age, we do have very few species. But then what comes to this symbiotic mycorrhizal fungi of this species, we do have well more of them. We do have 1,000, 2,000 something between the current estimates of the ectomycorrhizal species, and as you know that trees are totally dependent on them. But then I'm talking only about Norway spruce. Normally know that everyone knows that Norway spruce looks like that, beautiful, like white, like a normally shaped, but actually Norway spruce is genetically very divergent tree. There's lots of heterogeneity and lots of variation, and here are a few examples of the mutations of Norway spruce, how it can look like from above ground characteristics. But the question what we have been working with a long time ago actually now is about what is the intraspecific variation of Norway spruce in below ground and in the roots, and in particular in ectomycorrhizal communities. And so I start from our older studies, but that's to put give the motivation and reason and like the background for the studies, but then I end up in our very very recent new results about that, what we have found out. But this like a branch of research what we have been doing at the first Forest Research Institute, and then look at is that everything started from that our tree breeders question that they have lots of clonal feed experiments, because this is economically very important tree is spruce, and it's like a bread, and that's why there's lots of field experiments, mostly about clones. And they had a clonal field experiment where 15-year-old genetic variants, more than 90 of them, they were all healthy and basically well growing, but still they had double the growth differences in the above ground height growth. So that's why they were classified fast growing and slowly growing ones. And the question was that what can there be similar differences in roots, and what is happening? Can it be due to the roots and below ground aspects, because nothing is known about those. And we were studying them and found that the fast-growing clones, which had double the height, had double the ectomycorrhizal species diversity. And slow-growing clones had a little bit more than half of the species, even though their roots were not that much smaller. So the question is that then, that is the high diversity of the fast-growing ones, is it the reason for the good growth, or is it just a result of the good growth? And to study that, then we've first question was to see that if there's like a genetic basis in the host tree that they are more eager to form ectomycorrhizal associations. And then because we were lucky to work with tree breeders, we then continued to study with the clonal and seed-derived material. So actually in most of the experiments I will tell you about later, then we were working with the tree material or seedlings, or like clonal rootlets, which future is known. So they don't have yet, they are small seedlings, they don't have yet any differences in their growth rate, but it's known that tree breeders know that there will be differences in their growth rate in future. So that if I say that this is fast growing, it means that they will be fast growing or slowly growing. But it was important that when we were studying them, they didn't have any growth differences yet. But so what we studied, there were several questions we wanted to pose, and because there are several potential reasons for that. But as I said, the first thing was that we wanted to know if there are if the fast-growing ones have like a genetic ability to take up more diversity. And how to do it is that then anymore is not like a 4 slow-growing and 4 fast-growing ones. That's not enough. Then you need much bigger material. And we were testing like the random selection of 55 genotypes of Norway spruce, and then we were inoculating them all and letting them grow in greenhouse for quite a long time, and then we find out that this ectomycorrhizal community composition is under the genetic control of a host tree, but it doesn't look like that it extends to the ectomycorrhizal diversity. And then, but of course, that didn't give us an answer that to differences in the ectomycorrhizal species composition, do they differences appear before or after the differences in growth rate? And that's, we then established an experiment I will tell you about today. And then, of course, I just mentioned that one possible explanation is that there are like a more efficient defense-related gene expressions, so that mycorrhizals are defended away. And we were looking at those hypotheses, but all the experiments we have been doing say that there's no systematic differences in the defense-related activities of the host. But then something else. When we were looking at these mycorrhizal species in the genetic backgrounds, we found that they did have like a systematic differences in their roots. And actually it looked like in many experiments that fast-growing families and clones seem to invest more to their roots in the very early stage. And actually, in the experiment I mentioned about the mycorrhizal diversities, the root characteristics were more strongly under genetic control of the host, and they also co-varied with the long-term growth rate. That they were more stronger than the mycorrhizal diversity, and that's why we focused our studies then to the roots. Is the root formation associated to the growth rate, and is soil affecting the root formation? So then we were doing many studies about roots and how they are their structures. And actually we made a hypothesis that because this root branching seemed to be under genetic control of a host, so if there might be an optimal root structure which might collect diversity in the forest soil, because we know that forest soil is patchy what comes to fungal, also bacterial, but fungal communities. So if getting like a diverse what comes to species diverse mycorrhizal community, that would mean actually that there's also functionally very diverse community, and which means good ability to uptake of nutrients, which might mean good growth. So we had this like a root-driven hypothesis, and how we were analyzing the roots was from small seedling is this kind of Windritzer analysis. And then we started this root studies by comparing fast and slow growing families, which didn't have yet any differences, shoot the root biomass. And then we were studying the architecture of the whole root systems. And results of this was that we had several experiments, and both experiments showed the same, that here's a picture from the first experiment 2018, where I present the results so that if you put the lateral root like a extended to the, it was in the x-axis, you can see, and then you can, if you then calculate the root tips or number of branches of roots along that lateral root, then it was like a systematically difference between the fast and slow growing origins, that the future fast growing ones, the root tips and the branches peaked up further away from the stem. So it means that they were more, seemed to be more explorative, the root structure of the future fast growing ones. So that might give some benefit in a heterogeneous, spatially heterogeneous forest environment. And that the same was we found in another experiment where we did have a different set of fast and slow growing families. And these seedlings, they were like seed derived, because the root structure of the clones is very different, because it's much more rough. But all these results are obtained from seed material, where the roots are really branchy and like a, let's say that like a normal structure. But then, because these experiments all were done in a homogeneous growing substrate and in even temperature in a greenhouse, we wanted to see is the picture same, and is this picture mixed when or blurred if we change the environment. So then we didn't have any more fast and slow, but in this experiment we did have 15 families, so actually a whole local population of spruce in southern Finland, and almost 700 seedlings. And then we were putting them into 2 different temperatures, warm and cool summer, and we used 3 local soils, spruce soil, and then mixed spruce and pine soil, and then agricultural soil. And we were measuring like a phenotyping the whole seedlings from roots to the stem. But if I put together the results concerning roots, is that roots seem to be really adaptive, and they seem to be really variable, and the root size and the branching and morphology varied and responded strongly to the treatments. And many of these were independently varying from each others. And roots actually seem to be more plastic than above ground traits of the seedlings. And this also, what this would mean also in practice is that if you are doing like experiments with spruce seedlings, then it's good to have like a wide genetic background of the in your whole tree seedling material, because they use genotype. The genotypes are like reacting to the environmental or soil change differently, so your results are depending on what kind of genetic material you have. Then we had another more recent experiment of this, and there we started so that we had like a spruce population, the same families growing in previous agricultural soil and previous forest soil, and then we were collecting cones from both of those trees, and then we were sowing the seeds and planting them again and growing them as small seedlings, and then we were comparing the seedlings, and shortly we did see exactly the same again, that the soil had a major impact on the shoot traits and root traits. And additionally, we found that in a way it is like a kind of epigenetic thing, that the origin of even though from the same like a spruce family, like a genetically 50% the same, but then where it has been growing, was it growing in agricultural soil or previous forest soil, then it affected on the progeny seedlings and their traits, but probably it is because of the different nutritional condition of the host tree, so that the seed has like a nutrient reservoirs and like that. This is not totally unique, so it has been found so also before, so it wasn't that strange. But anyway, still the major effect on roots by the soil. But then to the third question, I said it's about the mycorrhiza. Now we just, because this working with these forest trees, everything is very slow. And if I go back to the beginning where I started my my talk, was that we had these the original fast and slow growing clones, where we found this phenomenon. During those early years, we had taken the branches from those fast and slowly growing mother clones, and we had tried to root them, and we managed to get a decent amount of fruit. We wanted to have more, but we got some. And then we were outplanting them back to the forest to fish ectomycorrhizal fungi. And so we wanted to see, so we put the original clones back to their childhood and see and how they like collect the diversity, and do we get the same result as in the original study. And this, even though we were like planting them almost 200 kilometers more south than where the original ones were growing, we did see again differences in these mycorrhizal and root endophytic communities, even though we didn't yet see differences in the above ground biomasses. Of course, fungal species were largely different because the forest was different, but also we saw some consistent results. For example, this very less, like a more very low resource demanding ectomycorrhizal species like Tylospora, were also now more common in the fast growing species, and then slow growing, fast growing hosts, but then slow growing ones, they had more, for example, ascomycetose fungi. So conclusions. So the question that do differences in ectomycorrhizal and root associated species, do the differences in these communities appear before differences growth rate of the host appear? And the answer looks like that, yes, early colonization by root symbiotic fungal species seems to be associated to the later growth rate, so that this mycorrhizal species might have a big impact on the growth. And then the second questions related to root formation. It also looked like that the future fast-growing ones, they have signs of more explorative root structures before differences in their growth rate appear. And then what we also found that the quality of the soil has a big effect on the roots. So thank you. That was Mayan. When earlier we were talking about visiting Helsinki. So next summer, please, you are welcome to our conference in Helsinki, Ecology of Soil Microorganisms.
Thank you very much, Taina. Okay, we missed the last picture. Yeah. Do you have any questions? It's a long story. I mean, in time. Yes, Stefan.
Yeah, very long. I tried to squeeze it. Yeah.
Stefan.
Thank you very much. It's very interesting story and very interesting topics. So, do you know if there is any differences in root exudates between slow and fast-growing trees? Because these differences in exudate could also explain a more attractivity in the first step of the colonization, maybe. So do you have some data on that?
Very good question. actually we don't have data, but then I think in France, it was François Le Tacon, I think his group might have been studying this. We were looking at the, we have been looking at some enzymatic profiles years ago, but we didn't find any differences because of the, actually we didn't look at, but we didn't find any differences by the host. We were looking at the enzymatic activities, but they all filtered by the fungus. So fungus was affecting, ectomycorrhizal fungus was affecting more. So the differences in enzymatic activities between the hosts were becoming via the differences in ectomycorrhizal community shift. But we didn't look at the otherwise the enzymatic activities. But I think group of François Le Taconi, Indra, Nancy has been doing something. I can't remember now what was the outcome of this.
Because it could be quite an important point. And I have another question. Well, nothing to do with actually your speech, but it's more about the picture in your background. Is it Craterellus tubiformis in the background of your picture? Because it's a very good edible mushroom that we have also in France. So is it the good? Is it that? It's a Craterellus tubiformis you have in the background of your picture? Yes, okay.
We can't hear you.
Sorry, it's very common in Finland, and we are picking
Okay.
up like
Yes, good
our
point.
forests
I mean,
are full of those.
we should really come at this period because very good and very good edible mushrooms. So yeah.
Please welcome. It's the season is starting in September. Yeah.
Yeah. Unfortunately, we will come in February, which is not really a good period to get
Yeah.
nice, let's say, mushrooms.
Yeah.
Thank you very much.
Yeah.